All right, let's go. The oxygen ceiling. A geologist kneels in a Wyoming quarry in 1986, brushing shale dust from a thin black seam that runs through the rock like a scar, and what he's measuring will tell him exactly how little air you would have to breathe. Dr. Robert Berner of Yale, working with charcoal deposits trapped in Cretaceous sediment, reaches for his notebook and writes down a number he has to check twice. The charcoal tells him how completely ancient wildfires burned, and complete burns only happen when there's enough oxygen in the air to feed them. He calls his colleague over, points at the seam, and says nothing for a long moment because the math keeps producing a percentage that doesn't match anything in the modern sky. Berner's reaction wasn't excitement. People who study paleoatmospheres describe him pulling the sample closer to the window light, turning it under his thumb, looking for a reason to throw the number out. He couldn't find one. Geochemist Ian Glaspell, working similar data decades later at Royal Holloway, would describe the same instinct. Check the lab equipment first because the alternative is stranger than equipment failure. What none of them could explain away was the consistency. Sample after sample, the oxygen ceiling held steady at a level that would leave you gasping within minutes if you tried to adjust to it directly. Newspapers that covered the discovery ran headlines about dinosaurs breathing supercharged air, painting a picture of bigger lungs and easier living. The peer-reviewed literature said something colder. The papers described an atmosphere that wasn't gentler, just denser with a gas your body isn't built to absorb in those quantities, one that at ground level would have triggered oxygen toxicity symptoms in an unacclimated human within the first hour. The press wanted majesty. The data described a slow chemical injury. The figure was eventually folded into mainstream paleoclimate models, cited in textbooks, treated as settled. The number itself was rounded for simplicity in public-facing material, the rounding never formally corrected, and conference panels through the 2000s would footnote approximately consistent with prior estimates, without revisiting the original charcoal ratios directly. A few specialists raised concerns about regional variance in the samples. Those concerns appear in conference proceedings, not in the documentaries that followed. But the ceiling itself was never the whole problem. The atmosphere didn't just have more oxygen. It had a different relationship between oxygen and carbon dioxide that no climate model fully reconciles with insect body size from the same period. And yet if you stood in that air for even 20 minutes, your own heartbeat would be the only sound competing with how loud your blood would feel moving through your skull. Bite force ceiling. A paleontologist in a Montana lab in 1996 lowers a fossilized jawbone onto a steel rig built to measure crushing pressure, and the rig's readout begins climbing past every number it was calibrated to expect. Dr. Gregory Erickson, then at the University of California, watches the gauge needle pass the threshold he'd built the experiment around and reaches over to recheck the bolts holding the jaw in place, assuming something has slipped. Nothing has slipped. He calls his lab technician over, has her read the display independently so there are two witnesses instead of one, and she reads back the same number without hesitation. Erickson's reaction, by his own later account, was to stop the trial and walk the readout down the hall to a colleague's office before writing anything in his notes. He wanted a second machine to confirm it. The second test, run on a different rig entirely, returned a number in the same range. What troubled the team wasn't the bite force itself but the bone fragments recovered from fossilized dung nearby, fragments that showed the animal wasn't just killing with that force. It was deliberately grinding through skeletal structure as a routine part of feeding, something almost no living predator does as a matter of habit rather than necessity. Documentaries that aired in the years following described the bite as the most powerful of any land animal in history, full stop, no qualifiers. Erickson's published paper was narrower. The force measured was the most powerful reliably calculated from available fossil evidence, a category that excludes anything without enough intact bone to test. The public got a record, the literature got a boundary. The bite force figures were incorporated into university coursework within a few years, presented in lectures as a closed measurement, with a brief mention that newer specimens may revise the upper bound tucked into footnotes that rarely made it into slide decks. A 2017 follow-up study quietly increased the estimated range, and the revision was announced in a short statement with no accompanying press conference. But the force measurement only tells you what the jaw could do once it closed. It says nothing about how an animal that size located, tracked, and closed the distance on prey fast enough to use that bite at all, a sequence biomechanical models still can't fully agree on. And yet the jaw alone, set on a table in a museum basement, is long enough that you would need both arms outstretched to span it tip to hinge. The crater floor. A drilling crew off the coast of Mexico in 1990 pulls up a core sample containing a layer of glass beads no wider than grains of sand, and the man holding the tube doesn't know yet that he's holding the exact moment everything changed. Geophysicist Alan Hildebrand, working with Glenn Penfield's earlier seismic data from the 1970s, lays the sample on a lab table in Houston, and starts separating the beads with a pair of tweezers under a desk lamp. He calls in a colleague, slides the dish toward him, and asks him to look at the shape of the glass without telling him what it is first. The colleague picks up the dish, tilts it toward the light, and goes quiet. What Hildebrand was looking at wasn't a story about an impact. It was a story about scale. He pulled the gravity anomaly maps Penfield had generated years earlier and laid them next to the tektite data. Geologist Walter Alvarez, hearing about the overlap, flew down to compare notes in person, and people in the room that week describe him standing at the map for almost 10 minutes without speaking. What none of the models could initially explain was why the debris layer above the structure was thin in some places and remarkably thick in others, as if the sky had emptied unevenly. The press ran the story as confirmation: the asteroid that killed the dinosaurs finally found, case closed. The geological surveys filed in Mexico and the US described something more careful: a structure consistent with an impact event of the relevant age, language that left room for revision. Where the headlines said certainty, the survey filings said probability. The crater was formally named Chicxulub, registered, mapped, and closed to further drilling by the early 2000s pending environmental review that was extended twice and never fully completed. A press conference announcing the naming ended early when a question about the debris layer thickness discrepancy went unanswered, and the moderator moved to closing remarks. The file sits open in name only. And yet the debris thickness problem was never solved. The official model accounts for the impact, the dust, the firestorms that followed within hours across entire continents, but not for why some excavated layers show signs of two distinct deposition events separated by an unclear span of time. Stand at the rim of that crater today, buried as it is under 100 feet of limestone, and the ground beneath your boots still stretches wider across than most people would care to walk in a single afternoon. The reflectance curve. A biologist in a fossil feather lab outside London in 2010 angles a microscope toward a sliver of preserved tissue thinner than a fingernail, adjusting the light until the structures inside resolve into rows. Dr. Jakob Vinther at the University of Bristol leans into the eyepiece and goes very still, then waves over a graduate student without looking away from the lens, telling her to look before he says anything that might bias what she sees. She looks, straightens up, and says she sees the same rows he does: pigment-bearing structures called melanosomes, packed in a pattern that only appears in modern animals with a specific, vivid coloration. Vinther's team cross-referenced the melanosome shapes against a database of living bird feathers, and the match wasn't approximate. Paleontologist Richard Prum at Yale, brought in to review the comparison independently, reportedly asked to see the raw microscope slides rather than the published images, a request colleagues noted because it signaled he didn't fully trust the figure as printed. What none of the reviewers could explain was why certain feather samples from the same specimen showed melanosome densities inconsistent with any single coloration, as if the animal carried two incompatible color patterns on different parts of its body. News coverage described the find as proof scientists could now see true dinosaur colors, presenting it as a settled visual fact. The peer-reviewed paper described a coloration consistent with specific pigment patterns under specific preservation conditions, a phrase that quietly excludes specimens preserved differently. The picture book illustrations that followed dropped the qualifier entirely. The melanosome method became standard practice across paleontology labs within a few years, cited in dozens of subsequent papers, treated as a reliable pigment key. A handful of researchers published cautionary notes about preservation bias skewing which melanosome shapes survive at all. Those notes are cited far less often than the original color reconstructions. But the preservation bias problem was never resolved, only acknowledged and set aside. The technique tells you what colors could survive fossilization, not necessarily what colors were most common in life, a gap the field still hasn't closed. And yet hold that fossil sliver up to a window and the color pattern preserved in it is vivid enough to read clearly from across a room, 100 million years after the animal wearing it stopped moving. The polar forest. A botanist working in Alaskan riverbank in 1985 chips frozen sediment away from a fossilized stump still rooted in place, exactly where it grew, and pauses when the rings beneath the bark don't match anything from a polar climate. Dr. Judith Topman Parrish, then with the U.S. Geological Survey, crouches lower, runs her thumb along the exposed growth rings, and counts them out loud to the field assistant kneeling beside her. The rings are wide, evenly spaced, showing no sign of the stunted growth that cold-adapted trees produce. She has the assistant count them independently, away from her own tally, and the two counts match. Parrish's team began excavating outward from the stump and found more of them, an entire buried forest standing upright, trunks spaced the way a living woodland spaces itself. Geologist Robert Spicer, examining similar polar deposits in Australia around the same period, reportedly stopped a field crew mid-excavation to resurvey the latitude markers, convinced the team had misread their own coordinates. They hadn't. What the rings couldn't explain was how broad-leafed trees survived months of total darkness every single year without the metabolic shutdown modern polar plants depend on to make it through. Wire coverage of the discovery described tropical forests at the poles, conjuring palm trees and humidity. The geological papers described a temperate, not tropical deciduous forest adapted to a seasonal light cycle far more extreme than any forest alive today experiences. One version implied warmth everywhere. The other described a narrower, stranger kind of mild climate built around total darkness for a third of the year. The polar forest findings were incorporated into Cretaceous climate models by the early 1990s, with the months-long darkness detail occasionally dropped from summary materials because it complicated the warm Cretaceous narrative editors preferred. A museum exhibit built around the find in the following decade quietly removed the darkness detail from its central placard after visitor surveys reportedly found it confusing. But no model has fully explained how a deciduous forest's internal chemistry adjusted to round-the-clock blackness for months, and then round-the-clock daylight for months after that, a swing no temperate forest manages today. And yet standing among those preserved stumps, still rooted in their original positions, the spacing between trunks is close enough that you could touch two at once with your arms spread only shoulder-width apart. If you want to see more videos like this, click the video on screen now and make sure to subscribe. The Acid Sea. A marine chemist runs water samples through a titration rig aboard a research vessel in the Gulf in 1998, watching the pH needle drift lower than the calibration curve printed for Cretaceous ocean conditions. Dr. Lee Kump, then at Penn State, rechecks the calibration sheet against the rig itself, assuming the equipment has drifted out of spec during the voyage. He recalibrates against a known standard, runs the sample again, and gets the same low reading. He hands the printout to a crewmate without comment and watches him read it instead of explaining what it means first. Kump's later published work traced the acidity to a specific volcanic pulse, and the timing lined up almost exactly with a known extinction pattern in shelled marine organisms from the same layers. Paleoceanographer Andy Rigwell, reviewing similar date years later, described checking the sediment dissolution patterns against multiple independent core sites because a result that clean made him suspicious of contamination. The pattern held across sites separated by hundreds of miles. What none of the models could account for was a narrow band of fully formed shells preserved intact within the same acidic layer, organisms that should have dissolved under those conditions but didn't. Science journalism described the period as a mass die-off, full stop. The peer-reviewed reconstruction described a die-off concentrated in specific taxonomic groups, with other shelled organisms in the same water surviving the acidification through mechanisms still under investigation. The public got extinction. The paper got a selective filter nobody could fully explain. The acidocean event was added to the standard Cretaceous timeline within a few years, presented in textbooks as a clean cause-and-effect chain. A conference panel addressing the surviving shell anomaly in the early 2000s ran short on time, and the question period was cut to allow for the next scheduled session, with no follow-up panel ever convened on that specific topic. But the survival mechanism in those intact shells has never been identified, only documented as an exception to an otherwise consistent rule. And yet drop one of those preserved shells into a glass of water today, and it will hold its shape and weight exactly as it did when it was still attached to something alive. The migration floor. A paleontologist surveying a dried lakebed in Texas in 2007 walks a quarter-mile trackway pressed into ancient mud, counting footprints that don't break stride once across the entire distance. Dr. Martin Lockley, then at the University of Colorado Denver, kneels at the clearest print, lays a measuring tape across it, and calls out the stride length to a student writing numbers into a field log. He has the student repeat the measurement on 5 separate prints along the trail without telling her the first number to rule out unconscious bias in her readings. Her 5 measurements match his within a fraction of an inch. Lockley's team mapped the trackway's full length and found it ran in a straight line for the entire surveyed distance, with no wandering, no pauses, no overlapping prints from feeding or resting. Geologist Anthony Martin, reviewing similar trackways from a separate Cretaceous site, has described the sensation of standing at the start of one and being able to see, without moving his own feet, exactly where the animal was looking the entire time it walked. What the trackway couldn't explain was the complete absence of any predator tracks crossing or following the same line, despite known predator activity in the same rock layer. Press coverage of the find described a dinosaur highway, a phrase that implies regular, repeated use. The published trackway analysis described a single, one-time movement event preserved in a single mud layer, with no evidence the route was ever used again by anything. The headline implied a road. The paper described a single afternoon never repeated. The trackway was preserved under a protective covering and opened to limited public viewing the following decade, with interpretive signage describing it in the highway language the original papers never used. A proposal to correct the signage stalled in committee and was last noted as pending review in records from several years ago. But no explanation accounts for why a single, undisturbed, predator-free trail exists at all in a landscape otherwise dense with competing tracks from multiple species. And yet walking that quarter-mile trail today takes a visitor as long on foot as it apparently took the animal that made it, stride for stride, without either of them breaking pace. The Amber Witness. An entomologist in a Burmese amber workshop in 2016 holds a polished resin chunk up to a desk lamp, rotating it slowly until something trapped inside catches the light at the right angle. Dr. George Poinar Jr., at Oregon State University, stops rotating the piece and brings it closer to his face, then calls his research partner over and asks her to look without telling her what she's looking for. She takes the piece, tilts it the same way he had, and after a moment says she sees it too, a feather, fully intact, attached to a small piece of tissue that is unmistakably part of a tail, not a wing. Poinar's team ran the amber through scanning analysis to confirm the tissue wasn't a contamination artifact from handling, and the scans came back clean. Paleontologist Ryan McKellar, brought in to examine the specimen independently, has described checking the resin flow patterns around the tissue multiple times because a sample that well preserved this deep in the find usually indicates disturbance, and he wanted to rule that out before saying anything publicly. What the tissue couldn't explain was a feather structure that matched no known modern bird and no previously cataloged dinosaur species, sitting in a category that didn't yet exist on the day it was found. News outlets described the specimen as a dinosaur caught in the act, frozen mid-motion. The peer-reviewed description was narrower, describing tissue attributable to a small theropod based on structural comparison, a phrase that leaves the exact species formally unresolved. The headlines named an animal, the paper named a category. The specimen was acquired through a private amber dealer before formal excavation records could be established for its original site, a gap noted in the supplementary materials and rarely mentioned in coverage. Export restrictions enacted in the region afterward made it functionally impossible to verify the original extraction site at all, a fact the paper discloses in a single sentence near its conclusion. But without a verified original site, no one can confirm what else was buried alongside that tail or what killed it. And yet that feather, sealed in resin for tens of millions of years, still catches light exactly the way fresh feathers do under the right lamp, with no decay visible to the naked eye at all. The pressure wave. A geophysicist reviewing seismic recordings from a Cretaceous boundary research site in 2004 watches a waveform spike well past where the chart was designed to register anything. Dr. Sean Gulick at the University of Texas pulls the chart from the recorder and lays it flat on the table, smoothing the curled edge so the spike is fully visible, and calls a colleague over before adjusting any settings. The colleague leans over the table, traces the spike with one finger without touching the paper itself, and asks whether the recorder had been reset recently. It hadn't. Gulick's later modeling, built from drill cores taken directly from the Chicxulub crater itself, indicated the wave wasn't a single pulse but a layered sequence. The first wave followed by at least one more within minutes. Geophysicist Joanna Morgan, co-leading the same drilling project, has described the moment the core samples confirmed multiple distinct shock layers as one where the team stopped discussing timelines and just kept reading the core, layer by layer, without speaking. What the layered shock pattern couldn't explain was why one specific rock stratum showed no shock deformation at all, sandwiched directly between two layers that showed extreme deformation on either side. Television specials described the impact as instantaneous, a single moment of annihilation. The drilling project's technical papers described a sequence of distinct, separated pulses occurring over a span of time long enough to be measured rather than instant, a sequence that complicates any single moment of impact framing. The instant became, on closer inspection, a precession. The undisturbed rock layer between the two shocked strata was logged, sampled, and shelved for future analysis, with no formal explanation issued for the gap. A planned follow-up study to investigate that specific stratum lost its funding allocation the following cycle and was not refiled. And yet the undisturbed layer remains exactly where it was found, untouched by forces that obliterated everything immediately above and below it. A silence preserved between two catastrophes close enough to step over with one stride. The Final Hour, a sedimentologist examining a fish fossil bed in North Dakota in 2019, traces a thin line of debris embedded directly in the gill tissue of a perfectly preserved specimen, and stops when he realizes the debris isn't local sediment. Dr. Robert DePalma, then affiliated with the University of Kansas, lifts the slab closer to the light and calls his collaborator over before he says a word, pointing at the gill rakers without touching them. The collaborator bends down, looks for several seconds, and asks where the glass came from. Neither of them has an answer yet. The spherules lodged in that tissue match the chemical signature of impact debris from a crater over 1,000 miles away, meaning this fish breathed those particles in within the same hour the world ended elsewhere. DePalma's team cross-referenced the fossil layer against magnetic and chemical signatures tied to the impact itself, and the dates weren't close. They were exact. Geophysicist Mark Richards, brought in to review the sedimentary sequencing, has described laying the core samples end to end on a lab floor and walking the line twice before agreeing the deposition had to have happened within hours, not centuries, of the impact event everyone already knew about. What none of the team could fully explain was why this single site preserved that exact hour in such fine detail, when almost every other boundary site on Earth shows that moment as a blur spread across thousands of years of erosion and mixing. Press coverage described the site as a snapshot of the last day of the dinosaurs, language that suggested total certainty down to the hour. The peer-reviewed paper was more careful, describing the evidence as consistent with deposition within a geologically short window, a phrase that left room for specialists to disagree about exactly how short. Some colleagues reviewing the same data suggested the window could be longer than the paper implied. The headline gave a single afternoon. The literature gave a range that still hasn't fully closed. The site was partially excavated, partially preserved under tarpaulin for future seasons, and partially withheld from outside researchers pending additional study, a decision that drew quiet criticism from paleontologists outside DePalma's immediate circle. A planned joint excavation with another institution was delayed, then scaled back, then never fully resumed at its original scope. And yet the fish remain exactly as they were found, gills packed with debris from a catastrophe that started somewhere else entirely. The distance between where they died and where the impact occurred farther than most people will travel in their lifetime, collapsed down to a single afternoon neither species saw coming. From the oxygen that would have left you gasping, to the bite force that could break bone on contact, to a fish that breathed in the end of its own world within the same hour it happened somewhere else, every one of these entries points to the same unresolved fact. The Cretaceous wasn't simply more dangerous than now, it ran on different rules entirely. Rules your body was never built to survive even for an afternoon. If you want to see more videos like this, click the video on screen now and make sure to subscribe.